Thank you to the organisers for the invitation to take part in this symposium. It looks very exciting. I want to focus today primarily on a study that my lab completed recently, but I want to put this work into a sort of broader context of ion channels and computation and cognition and behaviour. And so what my group is interested in as a set of questions is how circuits in the brain carry out computations and how those computations are used to guide behaviours. And when we think about neurons in circuits and how they compute, a really critical property of a neuron is its excitability, which is of course determined by the ion channels that it expresses. And we've known for some time that there's a great diversity of ion channels in the mammalian genome. And this, to us thinking about the brain and computations in the brain, begs the question of what's this diversity good for? On the one hand, sort of an idea that's quite popular is that it could be sort of redundancy. It may be that we can think of sort of computations in the brain with really quite sort of simple single neurons that act like a Hodgkin-Huxley or an integrated fire neuron. They fire action potentials, they have a resting potential, and a spike threshold. According to this view, which is probably held by a lot, maybe even most systems neuroscientists, that this is sort of all the neuron does. It sort of computes by summing synaptic inputs, generating action potentials. And according to this view, all these ion channels are here because maybe if one gets mutated, say a sodium channel, another one can step in and kind of take its place. Now, a contrasting view is that this diversity of ion channels perhaps supports computational diversity. So rather than there being a single generic computation that all neurons carry out, that there are many different single neuron computations, and that this channel diversity supports different types of computation in different neuron types. And this is not a new idea. The evidence for this goes back to work in the 70s and 80s by Chuck Stevens and Rodolfo Linas and numerous other people. And this idea can even extend to the possibility that a single channel might have different functions depending upon its cellular context. So the diversity of functions could be even greater than the diversity of channels. And this is something I'll come to later in my talk. So we want to know what single neuron computations are, how they work, and what the roles of specific ion channels might be. And our focus for quite some time has been that the Hcn family of ion channels. And as many of you know, these ion channels mediate currents called hyperpolarization activated currents. So these are really remarkable, really unusual currents in that they activate quite slowly on hyperpolarization of the membrane, and they deactivate on depolarization. So this is in contrast to most other voltage-dependent channels. And also, very importantly, their voltage dependence of activation is such that they tend to be open at around the resting membrane potential of many neurons in the brain. So what this means is two things. First of all, it means that they can contribute to the resting membrane potential. And second, it means that small changes in resting potential can bring about relatively large changes in the fraction of open or closed Hcn channels. So they're very well placed to sort of modify integration of synaptic responses. So quite some time ago, I did work as a postdoc where we looked at different behavioural roles for Hcn channels, and we found when we made mice with a global deletion of the Hcn channel that there were quite profound motor learning impairments. And when we made mice with a forebrain restricted deletion, there were actually, rather than impairments, we found enhancements in spatial learning and spatial memory. Now today, I want to focus on the neural circuits that we've been trying to understand that might mediate these motor impairments. And we know that when we make mice with a forebrain restricted deletion of the channel, that we don't recapitulate these deficits. And this has led us to focus on circuits in the cerebellum and its associated pathways as candidates for mediating these roles of the Hcn1 channel in behaviour. So this is a sort of cartoon of the classic cerebellar circuit. The really important neurons to us are the Purkinje cells. which are the output neurons from the cerebellar cortex, and neurons in the inferior olive, which are the sole source of an input called the climbing fiber input, which is very, very important for cerebellar learning. It provides an instructive signal for synaptic plasticity at synapses onto the Purkinje cells. And we know that HCM1 channels are expressed in the cerebellar cortex. The mRNA is expressed by Purkinje cells, and we can label cerebellar cortex with antibodies against HCM1. And we know also that there's quite specific in the brainstem labeling of the inferior olive with antibodies against HCM1. So these two parts of the circuit both have quite strong expression of HCM1 channels. So very briefly, I want to focus most of my time on the inferior olive, but very briefly, Purkinje cells, well, what do HCM1 channels in Purkinje cells do? How do they contribute to motor behaviors? To address this, we made mice where we restricted deletion of the HCM1 channel to Purkinje cells. And here in an adult animal, you see that the H current IH is completely abolished. And this disruption or reduction of IH begins postnatally. So this is a postnatal sort of removal of the H current from these cells. And behaviourally, what we find is that this is a rotor rod task to assay motor learning. We find on the first day of training no difference between control and mutant animals, but over several days of training we find an impairment in the motor performance of these Purkinje cell-specific deletion animals. And this suggests to us then that this is data from a global knockout mouse where you see both an early and a late impairment. And this suggests to us that the Purkinje cells seemingly are important for this sort of late phase of motor learning, or HCM1 channels in Purkinje cells are important for this late phase of motor learning. So to get some insight into what might be happening in the Purkinje cell, what we can do is prepare brain slices and make patch stamp recordings from Purkinje cells and activate their synaptic inputs. And I want to show you responses of the Purkinje cells to activation of inhibitory synaptic inputs. So the Purkinje cells spontaneously fire action potentials, and here we excite interneurons in the cerebellar cortex, and we see that we inhibit the Purkinje cell, we switch off spike firing, and we compare the responses of a control animal with a Purkinje cell deletion animal. And what we find is that the duration of this pause in spike firing is increased when we remove HCM1 channels. And we see this on multiple trials with spike rasters. You see here the delay until the neuron resumes spiking is increased. So what this suggests to us is that HCM1 channels are playing a role in integrating inhibitory inputs. And in particular, this fits very well with what we know about the biophysics of the current. As the membrane potential of the Purkinje cell hyperpolarizes, the HCN channels are activated, and here they provide a depolarizing current which helps quickly return the membrane potential back into the domain where the neuron can generate spontaneous action potentials. And this is a somewhat sort of conventional and reasonably predictable function of the channel based upon first principles from its biophysics and from what we know about the physiology of a Purkinje cell. So that's one important cell type in the cerebellar circuit. But what about these neurons in the inferior olive? These are an extremely intriguing population of neurons as an electrophysiologist. They have a number of really distinctive features which are not found in most neurons in the brain. So first of all, at rest, unlike most neurons in the brain, which will be silent at rest, many inferior olive neurons generate sort of subthreshold oscillatory activity at about 10 hertz. It's really quite remarkable when you record this from a neuron in a slice. So this is really something quite distinctive and not seen in most other central neurons. They also have quite unusual action potentials. So as well as a fast sodium spike, these neurons generate a sort of a complex sort of shoulder following the sodium spike, which is mediated by voltage-gated calcium channels. So they have sort of a priori sort of quite interesting sort of biophysics. And there were a couple of studies that looked at what IH or HCM1 channels might do in these neurons, and they suggested in an early study that the HCM1 channels might pacemake this oscillatory activity. And then in a more recent study that they might be involved in something called membrane potential resonance, or making the neuron more responsive to inputs of a particular frequency. Now these studies, and indeed most work on the inferior olive, has focused on these really interesting excitable properties. And what struck us, though, and was really quite remarkable, is that unlike most neurons in the brain, where most of the work looks at how neurons respond to synaptic input, there were very few studies investigating how these neurons in the inferior olive actually respond to or integrate their synaptic inputs, because most of the work is on these really sort of unusual properties. So when we started to work on the inferior olive, we set up, or Derek Garden in the lab set up a number of preparations to study how inferior olive neurons might respond to their synaptic inputs. So in this preparation, he's injected a virus expressing channel rhodopsin into motor cortex, and this virus labels neurons that project down towards the spinal cord. This is a section here of brainstem, and you can see the corticospinal tract labelled here, but also you can see axon fibres in the inferior olive. So when Derek prepared slices containing the inferior olive and then with a brief light pulse activated the axons, what he recorded is these biphasic synaptic potentials. So an initial depolarization followed by a hyperpolarization. And in most parts of the brain where one sees a biphasic response, this second slow component is usually mediated by GABAergic inhibition, but in this case, blocking GABA receptors has no effect on the response, but blocking glutamate receptors completely abolishes it. So from these experiments and a number of other experiments, we think this is a direct response to activation of glutamatergic inputs. And we think that this slow inhibitory component is actually caused by recruitment of calcium-activated potassium channels, which then generate this sort of slow hyperpolarization that you see here. And this actually, we thought this was quite cool. This actually was predicted by a model published several years earlier from Chris DeZu's lab, where he sort of built a biophysical model, not just of a single inferior olive neuron, but also of adjacent neurons. And he incorporated a really interesting feature of these neurons, which is that they have sort of dendritic spines, which are connected to dendritic spines from other neurons by gap junctions. And in his model, where he had a sort of ion channels which he knew would be expressed in the olive neurons, when he delivered synaptic inputs to these spines, he sort of predicted that this sort of biphasic response would be generated. So it's quite pleasing that our data sort of corroborate the predictions of this model. So, knowing now a little bit about synaptic responses of inferior olive neurons, we can ask, what do HCN1 channels do? And again, we can activate synaptic inputs. And here we're using light to activate channel rhodopsin. And we see subthreshold biphasic responses, which, as we increase the light intensity, trigger these action potentials. And we can block IH pharmacologically, and we see a number of things happen. So first of all, you hopefully notice the biphasic response has disappeared. You still see the excitatory component, but not the inhibitory component. What we also find is that the membrane potential is hyperpolarized very substantially. Now this we'd expect if HCN channels contribute to the resting potential of the neurons. And we also find that the timing of the action potentials is completely modified. So we see now a much longer delay until the action potential is initiated. And we see that the timing of the action potentials is also very variable from trial to trial. So suggesting that the HCN1 channels are controlling both the subthreshold synaptic response and the timing and actually the waveform also of the action potential that's driven by synaptic spike firing. So a number of sort of possible mechanisms come to mind. We can imagine from DeZu's model that HCN1 channels could be acting in a number of different locations. They could be acting at the soma to control the somatic resting membrane potential. They could be acting out here at the synapses in the glomerulus of the neurons we're recording from, or they could even be acting across these gap junction synapses in other neurons, and they could be influencing what we record here by an indirect action from other electrically coupled neurons. So Derek did a number of experiments to try and sort of tease apart these different contributions, and first of all, focusing on the synaptic responses, what he did was to ask how sensitive the synaptic responses are to modifying the somatic membrane potential. So we'd imagine that if the responses are sort of affected by HCN channels at the soma, that simply restoring the somatic membrane potential to its control value would sort of reverse the effect of blocking HCN channels. So this is a control synaptic response from a control animal. As we hyperpolarize the membrane potential to the membrane potential after block or deletion of HCN1 channels, we see that the biphasic response actually remains. So this argues that it's not a sort of a response that's close to the soma. Similarly, when we take the knockout at its resting potential and we depolarize to restore to the control potential, we don't bring back the hyperpolarization. So we can't rescue this response by restoring the membrane potential. So this suggests then that this sort of effect on the synaptic potential could be quite distant from the soma. So next we wondered if this is happening out at these glomeruli, and possibly if Hcn channels in the neurons that we're not actually recording from could play a role. So to test this, Derek patched neurons using a patch pipette containing the Hcn blocker ZD, and so he could dialyze the cell that he recorded from and block the Hcn channels locally and test whether the biphasic response remains, and then bath apply ZD and block all of the Hcn channels in the preparation. And when he does this, we see that after break-in, in this case, there's a slight sort of drop in the amplitude of the response, but we see the biphasic component remains even after we restore the membrane potential. We increase the stimulus intensity slightly, and you see very clearly these biphasic responses are still generated. But then when we block with extracellular ZD all the Hcn channels in the preparation, you see that the biphasic response is converted into a more conventional depolarizing response. And here's just the data sort of overlapped and the population data. So what this suggests to us then is that there's a contribution of Hcn channels from acting across gap junctions from other neurons to control the waveform of the synaptic responses in the cells that we record from. And I won't show you the data, but when we apply gap junction blockers, we also abolish this hyperpolarizing component of the response, which also very much fits with this idea that there's integration happening within these glomeruli involving gap junctions between multiple neurons. Okay, so that's the somatic response. Very quickly, what happens to the action potentials? We can do a similar test manipulating the somatic membrane potential. Here in the wild type, we see that when we hyperpolarize the somatic membrane potential, it looks like the knockout, this hump on the action potential is abolished. Similarly, with the knockout, these are spontaneous action potentials. When we depolarize, we bring back the hump. And the same thing happens when we look at synaptically driven action potentials. When we hold the membrane potential at close to minus 50 millivolts in both groups of animals, we see the shoulder on the action potential. So this gives us then quite really, I think, unusual functions for Hcn channels in the inferior olive cell types. First of all, at the synapse, they seem to be acting in a sort of across the synapse sort of fashion to sort of enable synaptic responses to be biphasic, probably by setting the local membrane potential at the synapse so that other voltage-gated channels can generate this hyperpolarizing component. Whereas at the soma, again, they seem to be acting by setting the membrane potential, and this time to control the waveform and probably the timing of action potentials. So we wanted to know what the consequences of these functions could be for behavior, or at least for neuronal activity during behavior. And so we collaborated with Ian Duguid's lab in Edinburgh to make recordings from Purkinje cells in behaving animals as animals sort of run on a treadmill. So these are cell-attached recordings that we make actually from the Purkinje cell, and we can record from the Purkinje cell this complex spike response, which tells us that the climbing fiber input from the olive has been activated. And actually, very nice work from Mike Heuser's group has shown that the duration of this complex spike response reflects the duration of the complex spike waveform in the inferior olive. So these experiments done by Marlies Usland enable us to look at the pattern of complex spike activity and also at the waveform of the complex spike activity. And the most striking result is that when we look either during quiet wakefulness or during movement at the coefficient of variation of the interspike intervals, so how variable the timing of the action potentials are generated by these neurons, the spikes become much more variable in the absence of Hcn channels. In addition, during movement, the number of wavelets and the duration of the complex spike waveform is also reduced, and both of these results are consistent with our slice experiments and very much point towards Hcn channels being important for controlling the timing of activity in the inferior olive during behavior. So, just to summarize then two sort of sets of results. So in Purkinje cells, HCN1 channels are activated by hyperpolarization and oppose inhibitory input. So if we remove the channels, inhibitory responses become larger. But in the inferior olive, which is immediately upstream from the Purkinje cells, the HCN1 channels have a very different function. So they're actually required for the inhibitory component of responses to glutamatergic input. And they determine the timing and waveform of complex spikes, both ex vivo and in vivo. So we think this points to cell type specific computations and cell type specific roles for a single class of ion channel. In doing these studies, though, it's also made us sort of realize sort of a lot of the challenges that we face in really mapping ion channel function onto computations in the brain. And I want to just quickly highlight a couple of those challenges. And the first really is to do with computation. So we only really have a very hand-wavy idea about what the computations are that are happening in most circuits. And this is a real challenge to really define the computations that happen and try and attribute functions to specific ion channels. But if we're right that ion channel diversity is in part required for establishing specific computations, then ion channels are going to be really important targets that we need to manipulate in very specific ways if we're going to understand in a really mechanistic sense how computations work in the brain. Another challenge that we've encountered is actually the complexity of brain circuitry. So we often treat the cerebellum as being a sort of a uniform structure with Purkinje cells sort of all being the same everywhere and the circuitry all being the same everywhere. And this is simply not true. There are extremely sort of specific microcircuits within the cerebellum, and these have very specific inputs, very specific outputs. And very likely, if we want to understand computations and what ion channels do in computations, we're going to have to sort of record at the resolution of these circuits in behaviors that engage circuits in very specific ways. And we're working on this at the moment, not in the cerebellum, but in another part of the brain called the entorhinal cortex, but very much with the viewpoint that we have to define the circuitry at a molecular level, at a connection level, in order to understand the computations that the circuits are carrying out, and in order to test specific models for how ion channels might be contributing to computation. So I'd like to finish there. I'd like to thank the people who did this work. So the work on Purkinje cells is by Arianna Rinaldi, who now has her own group in Rome. Derek Garden did the work on the inferior olive patch clamp recordings. Marlies Uslan did the in vivo work. And the in vivo work was a collaboration with Ian and Marty in his lab. I'd like to thank our funders and thank you for your attention.
Thank you, Matt, for getting off to an excellent start. So do you have questions for Matt? Yes.
So is the HCN1 the only isoform expressed, or do they have a...
So there are 4 isoforms of the HCN channels. In the olive and in the Purkinje cells, when you delete HCN1, you pretty much lose the H current. So it does seem to be the major isoform.
Heteromerized, right? So you might have other isoforms without the HCN1.
You could.
But
We would, maybe this is a bit naive, we'd expect, and this is based on experiments in neocortex or hippocampus, where when you delete HCN1, there's a large residual current, so you lose about 2/3 of the current. And there we know that HCN2 channels are also strongly expressed, and we think that that's, maybe they do form heteromers in the wild type, but we think we can see the residual current. In the cerebellum or the inferior olive, we don't see a residual current. I guess it could be that there's still another subunit expressed, but it doesn't make it into the membrane without HCN1. We don't know. Yeah.
Yes.
Yeah, Matt, in the early studies you guys did in the full knockouts, HCN1 knockout was associated with changes in theta activity. Most people, when they do EEG, actually reference the cerebellum. I'm wondering if you think that some of those changes were associated with changes in cerebellar activity, or in the frontal knockout, do you really see changes in midline theta?
So could the theta changes in the early studies be to do with the cerebellum? I forget where we referenced in those studies. I don't think so, for a couple of reasons. You see this in the forebrain specific knockout, so that argues against the cerebellum. There's also some very nice recent modelling work using detailed compartmental models to look at the field potential that actually predicts this result. So there's a certain sort of biophysical consistency now with what we found, which is quite reassuring.
Nikita.
Do you have Hcn channels in motor neurons?
In motor neurons.
This is the global knockout. At least in the afferents, there are Hcn channels, so your reflexes can be optimized this way. But if it's in motor neurons, so then it's even more so.
There might be. I'm trying to remember. I think Jim Dukas had a paper looking at this some time ago. So there certainly could be, and they could be important. We found that we didn't find a difference in sort of pre-training motor performance. And that sort of led us to think that, the stuff in the spinal cord might not be as important as what's happening in the sort of classical plasticity circuits. But that doesn't mean that there isn't some important function.
Just that in the global knockout, the difference was straight away from the start, if I interpret well this figure.
It depends on how you do the experiment. The rotor rod experiment we've discovered is not a single experiment. There's lots of different ways of doing it that produce slightly different results, but it can be there at the beginning depending on how you set the rotor rod up. Yeah.
Stephen.
Yeah, lovely, Matt. One thing I was wondering, a lot of labs, including my own, are always confounded by adaptive regulation. Are you lucky? And it's probably related to isoforms as well, but with us it was a completely a different ion channel could take over. Do you ever see any adaptive regulation in your system?
We don't, and we've looked quite carefully. I think there is evidence in neocortex for adaptive regulation of inhibition. Doug Bayliss' lab had a very nice paper some time ago in neocortex. I think partly the reason you may not see adaptive regulation for HCN1 channels is there's no other channel with the same biophysics. So you actually can't compensate HCN1 because there's nothing else that does the same thing. Also, you don't die if you lose it, so it's not super important for staying alive. So
Okay, on that note, we shall move on. Thank you very much, Matt.